Hello everyone, this is a tutorial on how to search and retrieve sequences from Ncbi. But before we dive into that, we need to understand why we are doing this and some of the fundamental concepts. Firstly, what is a biological sequence? Biological sequence refers to the linear one-dimensional ordering of nucleotides, the ACGT in DNA, or the ACGU in RNA, or amino acids in proteins. These molecular sequences serve as the fundamental blueprints for life, storing the hereditary information that dictates the structure, function, and development of all living organisms. Why do we retrieve sequences? We retrieve sequences to compare organisms, understand diseases, discover drug targets, trace evolutionary history, or predict protein function. All of this without having to sit at the bench in the lab or touch a single test tube. This forms the core of bioinformatics and molecular biology. Having established this, what is Ncbi? Ncbi stands for National Center for Biotechnology Information. It is the world's largest public biological database hub. It hosts dozens of sub-databases, and you need to know which one to use and why to use it. There are 5 core databases you'll be using in this course, and I'll be walking you through them one by one. Number 1 is nucleotide. It contains DNA and RNA sequences, and we use it when we are looking for a gene, mRNA, or genome region. This nucleotide database holds 3 different collections inside it. The first one is GenBank, the second one is RefSeq, and the third one is EMBL. What are the significant things about these 3 collections? GenBank is where scientists submit their sequences. It contains the raw, unedited sequences directly submitted by researchers, and this can contain error, it can contain duplicates, or other preliminary data. The RefSeq is a curated and reviewed version by Ncbi. It is more like a non-redundant database where each sequence is already reviewed, annotated, and verified. You can think of it as the official copy, and the EMBL is the European equivalent. The second core database is protein. It contains amino acid sequences, and we use it when we are looking for protein sequences. The third one is gene. It contains gene records, the sequences, the literature, the metadata of a particular gene with annotation, and we use it when finding a gene across organisms. The fourth database is SRA. SRA is Sequence Read Archive. It contains raw sequencing reads, and we use it when downloading next generation sequencing experiment data. While the fifth one is PubMed. PubMed contains library, and we use it to check or cross-check what has been published or what we find in our sequence database. Take a break, pause for a minute, and having established all these core databases, I have the question to ask you personally before we proceed. If asked, which database would you use to find mRNA from human insulin? What do you think should be your answer? We've established nucleotide as a database, protein as a database, gene as a database, SRA as a database, PubMed as a database. So which database would you use to find mRNA from human insulin? We're looking for mRNA. Our answer is nucleotide. We established that it contains DNA and RNA sequences, and we use it when we are looking for a gene, mRNA, or genome region. You can pause this video, go back, listen to the part of the 5 core databases, and understand which one to use and why to use each one. Before we dive deep into the practical aspects, another key concept I want you to familiarize with is ascension number. Every sequence in Ncbi has a unique ascension number. You can think of it as a library ID or a call number. It tells us where to find something and what kind of thing it is. And once you learn to read this, we can decode what something is before we even open it. For instance, if you see NM _00546 The NM tells us it is a RefSec messenger RNA. It is already created, reviewed. The number after the underscore, the 6 figure, before the decimal point, is the record ID. The decimal point and the number after it indicates the version. In this case, we can say this is version 2, meaning it has been updated. If this number were to be 1, it is the first version. It being 2, it has been updated to version 2. Imagine it is 3, it has been updated to version 3. NP means RefSec protein. NC means RefSec chromosome or complete genome. And this AF or any other 2-letter prefix followed by 6 digits is a gene bank submission. A gene bank submission is the one where the author submitted it and it has not been curated. An important distinction is that RefSec is anything starting with 2-letter prefix like NM, NP, or NC, and gene bank submission can be any other thing. When in doubt, I implore you to use RefSec because it is the most preferred, because it is already curated and it is more reliable. Now, let's dive into the practical aspect of all of this by retrieving the most famous gene in cancer biology, the TP53, the tumor suppressor. And as a plus, I want you to read on this and find out why this is biologically important. The first thing to do is to open your browser. I have opened my browser. Go to the Ncbi website, ncbi.nlm.nih.gov. Welcome to the Ncbi website. Familiarize yourself with the page, check out the things you see, read through before we proceed. This is an hands-on practical session, so I will implore you to open your laptops, open your PC, open your desktop, and let's practice along. We are looking for TP53, the tumor suppressor, and in the database drop-down, select nucleotide. And in the search box over here, type TP53, open a square bracket, that is the gene name, close the square bracket, and Homo sapiens, open another square bracket, that is the organism, close the square bracket, and mRNA, open a square bracket, that is the filter. We wrote this query this way, using all these feed tags in square brackets to tell Ncbi exactly which feed to search to reduce the noise. If not, a paper with TP53 might just come up in our results. You can play around this, check the result for this particular search, and compare it with another one without the feed tags. After typing your search query, click on search and expect the result. Now that the result is up, you can pick an entry of your interest and study the records. We have 1 out of 20 or 1 to 20 out of 197 results on this page. That is to tell us that we have about 197 results, so you can go through everything and pick an entry of interest. Exploring these results, you can outrightly see the base pair, linear mRNA. You can see the ascension, where they establish ascension numbers. So this already tells us that this is a gene bank record. NM tells us that this is a RefSec record for mRNA. Scrolling through it, exploring the pages, you can go to the next page and see for yourself more results and My interest is this, P53. There are a lot of options for it, but I'm particularly interested in this variant one. The first variant, this is a partial CDS, and we're looking for complete. So let's click this. We're waiting for the result to come up so that we can explore and study the result. Now that the result is up, you can see Homo sapiens tumor protein P53, transcript variant 1, mRNA. Ncbi reference sequence is this, that's the essential number, NM, RefSec, mRNA, the record ID, and it is a version 6. At the top, you see the locus line, which is telling us and showing us essential number, the length in base pairs, 2512 base pairs, the molecule type mRNA. We can see that it is linear, and we can also see the date that it was lastly modified. The definition is telling us about what we are looking for, ascension number, the version, keywords, source organism, Homo sapiens, reference author, title of journal, PubMed remark. Keep scrolling and studying the record. Scroll down, scroll down, scroll down, scroll down, scroll down. You can see a summary report. You can see primary. Completeness is full length. You can see the features. It's telling us the source, the gene, exon, misfeatures, exon, CDS. CDS is the coding sequence, the part of the mRNA that actually translates into protein. We can also see start and stop codon over here, the start and stop position. This is the codon start. Scroll through, 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 read through your record, understand your record. Go as far down as possible and study everything you see all over here. Further down, further down. And over here is the origin. This is the raw sequence itself. The ATGC, CTCAA, all of the sequences are here. And this is what we are going to export. And after carefully studying this record, we're at the bottom, you can scroll back up all the way to the top and click this FASTA link just below the title. FASTA. The FASTA format is up. This format is what we'll be using in almost every of our downstream tool and process. It is the most universal sequence format in bioinformatics. And looking at what the file looks like, you can see this arrow symbol just here. That is the header. It contains the ascension number and the description. And everything after this is the raw sequence. So when you check through and the check is done, come to this side and click send to. Ensure that you are clicking the complete record. Choose destination to be file. Ensure that the format is FASTA and click create file. You have just successfully downloaded your FASTA file. So from this single file, you can run a blast search. You can align it against other species. You can build a phylogenetic tree. You can hand it to a protein structure prediction tool. And this retrieval steps basically feeds every downstream process tool or downstream process in itself. So running you over the steps again, open your browser. Let's go back home. Open your browser. Go to the Ncbi homepage. Come to the whole database section. Click nucleotide. Go to your search bar. Input your query. Gene name and Homo sapiens, the organism, and mRNA, the filter. And search. Let your result pop up. Pick your interest, your entry of interest, like we did earlier. We're going for the one we picked already. Click on it, let it pop up, study your record, find the locus, read through the records, click FASTA because that is the format we are working with. Study your FASTA record. The arrow indicates the header showing the ascension number and description. These are the sequences. Come over here, click send to. Ensure that you're clicking on complete record. Ensure that you're choosing your destination to be file. Ensure that your format is in FASTA and you create your file. And you have successfully downloaded your FASTA file again. I've established that this retrieval step feeds every of your downstream process. For the purpose of this tutorial, being a beginner level introduction, it will be limited to this sequence retrieval alone. For a more advanced level, we get to learn BLAST and FASTA fetch. BLAST is basic local alignment search, and it is the single most important algorithm in bioinformatics. We use it to find sequences that are similar to the query in our database or in a database. And for that, we get to go to the BLAST website on Ncbi and also use Python and BioPython knowledge. And at a more advanced level, we also learn about eDirect, SRA, and custom packages. This particular eDirect is Ncbi's command line toolkit, and it lets you build Unix pipeline directly against Ncbi databases. You don't need a browser, you don't need Python overhead, and you can read on all of this just to have advanced knowledge and prep your mind. But most importantly, go over the tutorial, practice as much as possible as you can with what you've learned, and I will see you in the next